So what I've decided to title this talk is how can we utilize N of 1 or small subject sizes to study rare neurological diseases. And so we've thought a lot about, particularly in TMS, but across things, about having large controlled trials. And whenever possible, this is ideal. However, there's a lot of challenges with using this as our only mechanism to try to learn things. And remember that in many cases, diseases such as Alzheimer's disease, Broca's, aphasia, etc., were actually discovered in relatively small cohorts that would have never been actually published before, but really just came from a sense of intellectual curiosity, cataloging what people saw, and then ultimately just reproduced. And it was a significant finding that drives our thinking today. For example, the role of the hippocampus in memory was really well defined by a single case. And so we also have to think is that not everything that's neuroscientifically interesting is going to have the ability to handle all of the rigor that we would want to put it through in our ideal world. And if you think about, even if we talk about something more common like depression or other stroke or other diseases, if you think about all the possible trials that you could dive up to treat or study all of the areas in the brain, this can get very quickly into the millions of individuals that you need in order to do even a basic study for all of these things. But we also know that when we talk about transcranial magnetic stimulation, where you hit in the brain matters. So while it delivers a field, the evidence is pretty clear that we need to be in kind of the right area. And so having a basic understanding of the likely anatomy is fairly important for having an idea of how we might treat it one day with the right technique. And so one of the thoughts and one of the things that Infinitome provides, among other things, is the ability to look at individual subjects, to look at the data in a qualitative way, and to begin to have some idea of where we should aim our research, accepting that some things may not really work for a randomized controlled trial of some kind or other things. And so the engine basically utilizes different aspects of machine learning with large amounts of normative data and some sophisticated statistical techniques to not only collect connectomes of structural and functional connectomes and do this in an automated way, but to also provide norms, or what are called anomaly matrices, to tell us what is not normal in the data that we're looking at compared to normal healthy adults. So I'm going to give an example of where we were able to look at a fairly rare condition that has been really poorly studied, and I don't see a path where this will ever have a randomized trial because it's so rare. But ultimately, we were able to at least shine some light into the black box and get an idea of potential mechanism, which, again, the next time a physician somewhere is encountered with this rare disease, they have at least some idea what to tell the patient, as opposed to this is really strange. So for those of you not familiar with foreign accent syndrome, what this basically refers to is a syndrome where somebody develops an accent that is not their native accent. And it's often sudden or very quickly onset. And essentially what's fascinating about this disease is that while people talk it with the accent, they don't use the idioms of the destination language. So in this particular case, or actually in these particular cases, these are young women who woke up with accents that were different than their native. So 2 were Australian to Irish, and 1 was American Latina English to British English. And so this has been described, it's often found in the setting of strokes, but what made these 3 things unique is that none of these people had structural damage to the brain. So if you look at the T1, is pretty unrevealing in all 3 of these cases. So the first A and B are people who went Australian English to Irish English. They don't use Irish idioms. They don't talk like Irish people, but they sound like Irish people, or at least very close to that. And in the case of the British English, American to British English, you can see something very similar. We also looked at the structural connectivity of the language system on DTI, and you can see that all of them have fairly structurally normal language systems in orchid fasciculus, frontal ascending tract, etc. all of which suggesting that unlike people who have brain tumors, trauma, or things like this, there's no history of this in any of these individuals. One of them was after a sore throat, and a couple of weeks later, that's the only thing that happened in the weeks preceding, woke up one day and has never gone back to Irish. Now, what you can look at when we begin to look at connectivity matrices and what's possible to do with this is we can look at the functional connectivity matrix. This is with resting state fMRI in these individuals, and begin to look at both the core language and temporal language areas, and begin to look at the raw correlations between those areas. So you can see in these adjacency matrices. We can do this for both language, the sensory motor system, and also the subcortical regions, including basal ganglia and cerebellum. But we can actually compare them against each other. So that's what you're looking at on the right, is language versus sensory motor and language versus subcortical. And that's interesting, but oftentimes these matrices can get quite big. And how do we know if any of this is actually interesting? So what we can then do is then look at this in terms of anomalies. And you can see that depending on the individual, they all have anomalies in both the language and sensory motor system. But when we compare them against each other, this is where it really gets pretty interesting. Many of them have abnormalities in how different parts of the language system talk to, say, the cerebellum, how they talk to the basal ganglia, how they talk to parts of each other. So if you look in this middle column, this is actually the most profound one. And you can see that not only are there inter-language parcellation functional connectivity outliers, and these are 3 sigma outliers compared to a cohort of about 350 normal adults, but also that we see decreased connectivity with these areas in multiple parts of the sensory motor cortices, as well as different parts of how these areas talk to the basal ganglia. So what we can conclude is not necessarily that there's an individual pattern, because there are differences in all of these people, but that oftentimes language and sensory motor functional connectivity anomalies can exist in the absence of structural damage to cause foreign accent syndrome. And of course, we can now begin to get hypotheses on what would be more interesting to study in the future. So again, while this isn't a definitive answer to what is the cause of foreign accent syndrome, or is there a cause, what really arises out of it is that without this kind of technology, it would be impossible to say much of anything about a cohort of 3 patients, even though this is an incredibly rare disease that was basically triggered by a news story on one of these patients, that the other patients came out of the woodwork to get their subs scanned because they were curious on what this showed. And we would have never had any tool that would have been able to identify anything objectively in a way that was scalable and learnable. But now we have some insight into what's actually happening. So one of the things we've have an interest in our company, we make FDA-approved products, we work with doctors, but we also have a very strong interest in contributing to the advancement of neuroscience generally and working with researchers. And one of the things that we came up with partnering with Brainbox is a way to break some of the challenges of bringing fMRI-based technologies into imaging research, into neuroscience research and TMS research more specifically. And our goal when we awarded this was to aim for providing the ability for early career researchers to have access to scan time, which is often a rate-limiting step. So after considering some very, very good submissions, we ultimately decided to award this to a group at Trinity College, specifically, I'm sorry if I butcher this, but Roisin McMackin and her mentor, Richard Carson. So congratulations. They'll be studying intermotor connectivity and inter-hemispheric connectivity, linking TMS and MRI-derived measures. And one of the things we were particularly impressed on is how they really had a meaningful, thoughtful process on how they would integrate functional connectivity and begin to link it to some of the neuroscientific findings that we could see in TMS. So congratulations.
So Roisin and Richard, if you are available, I think I should be able to hand over. Who owns the slides first? Who do I make?
I have the slides.
Ooh, okay, Roisin, you should now have the option to share your screen.
Okay, well, thank you so much anyway, first and foremost. We are really delighted to receive this amazing prize. So, I mean, we'll get into it now, but it's really a really important and unique opportunity for us to do this research. So we really appreciate it. So can you see those slides there?
Yes, all good.
Great. Okay, so as I said, Professor Carson and I were really delighted to get this this prize, and the project that we put forward is premotor to primary motor interhemispheric connectivity, integrating TMS and MRI derived measures. So the reason that we were interested in this is that we've been spending a while looking at TMS, or transcranial magnetic stimulation based measures of interhemispheric connectivity between the motor areas. So just as a basic background for anyone who's not familiar with this, many people will use TMS, or particularly paired pulse TMS, to investigate a measure called inter-hemispheric inhibition. So what this involves is applying a conditioning stimulus over one primary motor cortex, and then a test stimulus over the other primary motor cortex. And it's found that if the conditioning stimulus is applied at about 10 or 40 milliseconds before the conditioning stimulus, that it will have an inhibitory effect over the other cortices' ability to respond to the test stimulus. So this is then inferred to be the result of inhibitory signals passing along corpus callosum interhemispheric networks. So this measure of interhemispheric inhibition, or IHI, is, as I mentioned, typically measured between one primary motor cortex and the other. And it's been very commonly used to study the function of these motor-to-motor tracts in health, such as in bimanual coordination, but also in the study of diseases like motor neuron disease or stroke, in order to study or quantify how a disease impacts the interhemispheric networks that join the motor systems of the two hemispheres. So the reason that we are interested in branching out to premotor to primary motor interhemispheric networks is based on some work by Kathy Ruddy and Richard Carson and their colleagues, but also, of course, other groups, which have been using structural MRI to look at the relationships or the structural connections between the motor areas of the two hemispheres. And you can see from this figure, just as an example, that of course there is rich connectivity between homologous regions of the two hemispheres. So between, for example, one primary motor cortex and the other. But what we also see is that there is robust connections between the premotor cortex of one hemisphere and the primary motor cortex of another, which you would expect given the role of the premotor cortex in motor planning. So particularly you can see here that this connectivity is most robust and most rich between the dorsal premotor cortex and the anterior primary motor cortex. So we wanted to look at the best way to study these connections functionally using transcranial magnetic stimulation. So with Glenn Calvert and Richard and myself, we were using a paired pulse dual coil protocol where the conditioning stimulus is applied over the premotor cortex of the non-dominant hemisphere, and then the test stimulus is applied over the primary motor cortex of the dominant hemisphere. And what we found was that the dorsal premotor to motor interhemispheric inhibition was expressed if we used a prostero-anterior coil orientation for the test coil, but not an anteroposterior coil orientation. And this was different from the primary motor to primary motor interhemispheric inhibition, which was present irrespective of the test stimulus coil orientation. So that was indicative that they do have distinct qualities and underpinning properties, but they both showed some commonalities. So, for example, both are greater with increasing conditioning stimulus intensity if you use the postero-anterior coil orientation. So I won't go into the detail, but the paper, by using various interstimulus intervals and various test stimulus intensities and coil orientations, enabled us to refine the parameters that we would use to study this measure to maximally induce this inhibition. And the idea behind this is that we can then use these optimized parameters to use TMS to study this connectivity. However, one of the major limitations is that we didn't have access to MRI scans for these people. So in order to determine where the dorsal premotor cortex was, we had to estimate this based on craniometric measurements over the scalp. So the premotor cortex was defined as being in a certain spatial location relative to the hotspot of the muscle in that hemisphere. And of course, that potentially means that we could have been measuring different functional subdomains of the primary motor cortex within those individuals, or potentially even engaging some of the some premotor and primary motor areas with the conditioning stimulus. So going forward, what we want to do is to map our stimulation-based measures using TMS of the callosal connectivity into individual brain connectomes using MRI, so that we can much more specifically determine where the conditioning is being applied. So, of course, this is where Infinitome comes in, and where this prize really provided the ideal opportunity for us to dive into this. So on the right-hand side here, you can see a figure from this paper by Glasser et al., where they define this atlas, which gives a much more detailed insight into the functional parcellations within different structures within the cortex. And you can see in the premotor cortex, there's actually 6 functional parcellations within that, which are shown to be differentially activated during different functions. So we want to know which of these contributes to the inter-hemispheric inhibition effect, and understand how the activation of these areas with a conditioning stimulus regulates the activity of the primary motor cortex in the other hemisphere. So what we plan to do is to scan 25 individuals, which will be funded by this prize, and undertake structural and functional imaging in these individuals. And then what we can do with Infinitome and with the support from the OAT team that we will receive as part of this prize, we'll be able to identify individual cortex parcellations of the premotor cortex and the primary motor area in these individuals. And with these individualized structural and functional maps that the Infinitome platform can generate, we can quantify the structural connectivity and the functional connectivity of these premotor areas to other motor areas, both within the same hemisphere and the other hemisphere. So that will enable us to get a really individualized and rich picture of the structural and functional connectivity within each individual, but it will also enable us to determine the coordinates within each individual of each premotor subdomain, which we can then export to the BrainSight neuronavigation software, so that we can apply TMS in a much more personalized and accurate method compared to using craniometric approximations of the premotor location. So after we do the imaging, we'll then get the same people back to do paired pulse dual coil TMS. So having generated the structural and functional maps of their connectome with Infinitome, we can then apply conditioning stimuli to the specific parcellations within the premotor cortex, using the interstimulus interval and conditioning stimulus intensity protocols that we previously refined, and then apply our test stimulus to the dominant hemisphere motor cortex with different coil orientations. And we'll use this threshold hunting protocol, which in summary means that we can see the inhibitory effect of the conditioning stimulus based on how much we need to adjust the test stimulus to get the same motor evoked potential amplitude when the conditioning stimulus is present versus when it's absent. So we expect that if the conditioning stimulus has an inhibitory effect, we'll have to use higher test stimulus intensities to override the inhibitory effect of the conditioning stimulus in order to maintain our desired motor evoked potential response. So this will enable us to quantify the relationship between structural and functional metrics of the individual's connectome and the TMS-based measures that they give of inter-hemispheric inhibition. So what we can learn from this, this is actually kind of quite a basic list, because this Professor Carson and I were just discussing earlier about how there's actually a huge amount of really interesting and exciting data that we can take from this. But just at the very top level, first of all, we can look at what structural and functional networks underpin this inter-hemispheric inhibition, which is really important because if we're going to use these measures to study malfunction of inter-hemispheric tracts and disease, we really need to be more certain about what that reflects in terms of underlying dysfunction and degeneration. So this could be changes in both intracortical and inter-hemispheric networks, which by using the functional data will really give us an insight into perhaps more indirect tracks between different areas, and that may also be responsible for the phenomenon, as well as more direct connections between the areas. And also it will help us identify how each functional subdomain within the premotor cortex regulates the primary motor cortex and can evoke this inter-hemispheric inhibition phenomenon. And as part of that, because we're going to recruit an age diverse cohort, we can look at how this connectivity changes with things like age. And I think it was really important what Mike said just beforehand about, this by using this personalized approach, eliminating the variation that's involved in more approximating protocol, and particularly, for example, in the case of diseases like MND or stroke, where it's not possible to engage, to really bring in huge cohorts of patients. So to be able to much more specifically and confidently target specific networks in smaller groups, we anticipate that this will enable us to have much more statistically powerful investigations of these networks in health and disease. And overall, this will enable us to have refined tools by which we can quantify premotor to motor network function in health and disease. But as I said, there's a huge and much more detailed and exciting level of analysis that we can undertake, and we hope we can report that to you in the coming months and years. So I'd just like to say so very much thank you to OAT and as well to Brainbox, and we really look forward to working with OAT and to this exciting project. So thank you so much on my own behalf and on the behalf of Richard Carson, who is here as well.